Imagine you need to analyze some DNA at a crime scene, for example, and you have some cells. Now imagine you could take those cells, break them open, pull out the DNA and say, alright, I'm going to analyze from there to there. And then you can just get rid of the other pieces of DNA, you just want that one section, and once you have that section, you can make millions, if not billions, of copies of it for further analysis. Well, you can stop imagining because that is very real. And those two processes are called PCR and Gel Electrophoresis. Let's go. So PCR, which is short for polymerase chain reaction. And if you paid attention at all in biology class, then you should know that any sciencey looking word that ends with an ACE is most likely going to be an enzyme. And that is indeed the case for polymerase. Now if you paid even more attention in biology class, then you should know that DNA polymerase is present when you are replicating DNA. And that is essentially what we're doing with PCR. PCR is taking a section of DNA, making lots of copies of it for further examination. So how do we actually perform PCR? Well, the first thing you're going to need to do is denature some cells. You do that by having some cells and putting them into some distilled water and heat that up to 95 degrees Celsius for about 5 minutes. So that's going to break open the cell and, more importantly, break apart the hydrogen bonds holding the two strands of DNA together, so you're left with single-stranded DNA. So now you have your solution with the single-stranded DNA, but you also have a lot of other things inside of that test tube that you will not be needing. So what do? You centrifuge it. Centrifuging is basically spinning it really quickly so that there's higher G-forces on the test tube. That's going to mean that the heavier little organelles and all the pieces that are inside of a cell are going to sink down to the bottom of the test tube, while the DNA, being lighter, will stay somewhat at the top. After that, you can extract the solution with just the DNA in it, and then you're ready to move on to the next step, which is adding primers. The way you should think about primers is as a puzzle piece. The primer is a custom-made puzzle piece of bases of DNA, and you're going to throw that into the solution so it'll attach to only one certain section of the DNA. It only attaches to the section on which it fits perfectly. So that means that you can basically trim off the rest of the DNA, because now you have one tiny section of now double-stranded DNA, whereas the rest is still only single-stranded. Now there's a couple more things that you need to add into the solution to make it actually be able to replicate itself. The first one being an enzyme that replicates DNA. Now, DNA polymerase might be the first thing you jump to. However, that is not a good choice, because DNA polymerase is great, but it's made to work at the temperatures that are present inside of our body. However, during PCR, we're going to be heating a solution to temperatures that are much, much higher than the ones in our body, which would essentially completely destroy the enzyme that we have, DNA polymerase. So what do? We take a field trip. So our scientists went to look at some geysers. And where would you find some geysers? Well, the Yellowstone National Park, of course. And what they did was they took a sample of the area around the geysers and found that there was some bacteria living there. What that told them was that there had to be some sort of bacteria that's able to replicate DNA even under very high temperatures. So the enzyme they found was called Taq polymerase. And Taq stands for this really long word right here, which is actually the name of the organism that lived close to the geysers. So now we have our polymerase that can work under really high temperatures, which is great. So the final thing we need to add to our solution are building blocks. Building blocks for polymerase, so bases, nucleotides. Once we have that inside of our little PCR test tube, which is a real thing, we are ready to throw that into a thermal cycler. A thermal cycler is a machine that is, although expensive, very good and very efficient at changing its temperature rapidly. And now what we're going to do is we're going to put our PCR test tubes into that machine and then set it to go on a cycle. Now the first temperature we're going to reach in this cycle is 95 degrees Celsius. Now if you remember from a couple of minutes ago, 95 degrees Celsius is the temperature at which the hydrogen bonds holding together our two strands of DNA break apart, giving us two single strands of DNA. So the second temperature we're going to try to reach is 55 to 65 degrees Celsius. This is the temperature at which the primers will anneal to our now single strands of DNA, creating two double-stranded strands of DNA. Now the third temperature we're going to try to reach is 75 to 80 degrees Celsius. That is Taq polymerase's optimal working temperature, which means that it's going to start to recreate those bonds between these strands of DNA, giving us two complete double-stranded strands of DNA. After this third step, we're just going to restart the cycle, and since we now have two double strands, those are going to break open, give us four single strands, and then we're going to complement those, let Taq polymerase do its thing, and then we're going to have four complete strands of DNA. Then we're going to split those again, we're going to have 8, again 16, 32, and then it just grows exponentially. So at the end of 30 cycles, you have millions and millions of copies of your DNA sample. Alright, now we have all the DNA we need. It is time for gel electrophoresis. So as the name implies, gel electrophoresis is probably going to have something to do with gel and electricity. So now the first thing you're going to want to do is make your gel. So the most common type of gel used for gel electrophoresis is going to be an agarose gel. Now agarose is a type of plant that is used to make the gel as opposed to gelatin, which is made from ground up bones. So what you're going to want to do is get 1% agarose and 99% water, heat those up and mix them together to create the gel. Then you're going to pour that into a square mold, making sure to add a comb in to create little wells to put our DNA in afterwards. The fact that we have a 1% agarose gel means that there's going to be tiny little microscopic pores inside of our gel, which are going to come in very handy later on. So once we've done that, we're going to have to wait for our gel to solidify. And once it's done so, we can go ahead and take our DNA solution and put it into those wells. The next thing we're going to do is also add a solution which will color the DNA so that we can look at it under UV light after we've performed the gel electrophoresis. So now we're almost ready to start gel electrophoresis. The only thing we have to do is put our gel into a buffer solution and then apply a current. Now an important thing to know about DNA is that due to the phosphate in its backbone, it will always be negatively charged. What that means is that it's always going to want to go from the negative electrode to the positive electrode once we apply a current to the gel. Now before we apply our electrical current to the gel, what we're going to do is we're going to want to add in a DNA ladder. Now what that is basically scientists made a solution with a bunch of different sized chunks of DNA that are going to separate throughout our gel, providing us with kind of like a scale to estimate the size of the DNA we're trying to analyze. So now we're going to go ahead and actually run our current through the gel for about 30 minutes, and that is going to give the DNA enough time to make its way through the gel. So after you're done with that, the next thing you're going to want to do is carefully take the gel out of the buffer solution, making sure not to break it, and then you can go analyze it under a UV light. And who knows, maybe, just maybe, you'll find yourself a criminal. So that's going to go ahead and wrap up this video about PCR and gel electrophoresis. I hope you enjoyed it. I hope you learned something, because this did take quite a while to make, so that you got something out of it. Other than that, I want to thank you guys so much for watching. Goodbye.